Titration experiments, importance of indicators in quantitative analysis. Titration experiments, importance of indicators in quantitative analysis. Introduction, 100 words. Titration experiments play a critical role in quantitative analysis, enabling chemists to determine the concentration of a substance accurately. One key element in these experiments is the use of indicators, substances that change color at specific pH ranges, signifying the endpoint of a titration reaction. In this article, we will explore the importance of indicators in quantitative analysis, highlighting their role in achieving accurate and precise results. We will delve into how indicators work, their types, factors affecting their selection, and their significance in titration experiment. TS. Section 1, Understanding Indicators, 300 words. Indicators are chemical compounds that undergo a distinct color change at specific pH values. This property makes them invaluable tools in titration experiments where the objective is to find the concentration of a substance in a sample solution. They provide visual cues to indicate when the reaction between the analyte and the titrant is complete, leading to accurate measurements. Indicators work based on the principle of acid-base reactions. They exist in different forms, depending on the pH of the solution. When an acid-base indicator is added to a solution, it undergoes a reversible reaction, and the equilibrium position depends on the relative concentrations of the acid and its conjugate base. At different pH values, these forms have distinct colors, enabling chemists to visually determine the endpoint of a titration. Section 2, Types of Indicators, 400 words. Indicators come in various types, each suitable for different pH ranges. Some of the commonly used indicators in titration experiments include phenolphthalein, bromothymol blue, methyl orange, and Li. Phenolphthalein, for instance, is an example of a colorless acid that changes into a pink color in a basic environment. Its transition range is around pH 8.2 to 10.0, making it ideal for titrat. Ions involving strong acids and strong bases. On the other hand, methyl orange is a dye that shifts from red in acidic solutions to yellow in alkaline conditions. It is particularly useful in acid-base titrations where the endpoint coincides with the pH range of methyl orange. This makes it suitable for titrations involving weak acids and strong bases. It is important to note that the selection of the indicator depends on the nature of the titration being performed. For example, if the sample is an acidic solution and the titrant is a strong base, indicators with transition ranges around the neutral pH region, such as bromothymol blue, would be ideal. This ensures a clear color change at the equivalence point, enhancing the accuracy of the measurement. Section 3, Factors Influencing Indicator Selection, 400 words. Several factors play a crucial role in selecting the most appropriate indicator for a titration experiment. One such factor is the pH range of the indicator. The selected indicator's transition range should correspond to the pH at which the endpoint occurs for the specific titration. This ensures that the color change is distinct and easily observable. The concentration of the analyte and titrant also affects indicator selection. If the solution to be titrated has a low concentration, indicators with a sharp and narrow transition range are preferred. This enables the chemist to pinpoint the endpoint precisely. In contrast, when the solution's concentration is high, indicators with a broader transition range are suitable as they can accommodate Gradual color changes. Additionally, the compatibility between the indicator and other chemicals in the solution is crucial. Some compounds can interfere with the indicator's performance or produce undesired color changes. Therefore, it is essential to consider the chemical compatibility of the indicator with other substances present in the solution. Section 4: Significance of indicators in titration experiments, 400 words. Indicators play a critical role in achieving accurate and precise results in titration experiments. They provide a visual signal of the endpoint, allowing chemists to stop the titration at the right moment, leading to more accurate measurements. Without indicators, it would be challenging to determine the equivalence point precisely, as the naked eye cannot perceive subtle changes in the color. SS solutions often encountered in titrations. Moreover, indicators help save time and resources by preventing overtitration. Overtitration can occur when the titrant is added in excess, resulting in inaccurate measurements. Indicators, by indica, ting the endpoint, help chemists avoid overtitration, ensuring that only the required amount of titrant is added. Section 5: Future developments in indicator technology, 300 words. As technology advances, there are ongoing efforts to develop new indicators and improve existing ones to further enhance the accuracy and efficiency of titration experiments. One area of focus is the development of indicators that can provide more precise and distinct color changes, allowing for even greater accuracy in determining the endpoint. Researchers are also exploring indicators that are more resistant to interference from other substances in the solution. This would reduce the risk of undesired color changes and ensure that the indicator performs optimally in various chemical environments. Furthermore, there is a growing interest in the development of indicators that can be detected and interpreted electronically. This would eliminate the subjectivity associated with visual observation and provide precise measurements of the endpoint. Electronic detection methods, such as spectroscopy, can offer real-time monitoring and accurate determination of the concentration of the analyte. In addition to advancements in indicator technology, there is a focus on developing automated titration systems. These systems would incorporate advanced indicators and electronic detection methods, allowing for more efficient and precise titration experiments. Automation would also eliminate human error and improve the reproducibility of results. Another area of future development is the exploration of indicators that can be used in titrations involving non-aqueous solutions. Traditional indicators are designed for use in aqueous solutions and may not perform optimally in non-aqueous environments. Developing indicators specifically for non-aqueous titrations would broaden the scope of quantitative analysis, allowing for more versatile applications. Section 5: Future developments in indicator technology, continued. One area of future development is the exploration of indicators that can be used in titrations involving complex samples. Many applications require the analysis of samples that contain multiple analytes or components. Developing indicators that can selectively react with specific analytes or components would enable accurate and reliable measurements in complex sample matrices. Moreover, researchers are investigating indicators that are more environmentally friendly and sustainable. Traditional indicators may contain toxic or hazardous substances, posing risks to human health and the environment. By developing indicators that are non-toxic and biodegradable, chemists can ensure safer laboratory practices and reduce the negative impacts on the ecosystem. Additionally, There is a growing interest in the integration of indicators with microfluidic systems. Microfluidics technology allows for precise control of fluid flow and reaction conditions on a SM all scale. By incorporating indicators into microfluidic devices, chemists can achieve faster and more controlled titration experiments, leading to higher throughput and improved efficiency. Furthermore, the development of indicators with enhanced stability and shelf life is another area of focus. Traditional indicators may degrade over time, leading to inaccurate results and the need for frequent replacements. By designing indicators with improved stability, chemists can reduce the frequency of indicator preparation and ensure consistent and reliable performance. Lastly, there is a need for indicators that can be used in novel titration techniques and experimental setups. As new methodologies and instruments emerge in the field of quantitative analysis, indicators must adapt to these changes. Section 6, Conclusion, continued.